Okay, hello everyone. So this is going to be a lecture for your labs 25, 26, and 27. And it will kind of help you go through your lab report and hopefully explain some of the major concepts in each of these labs. So let's start off with the muscle system. So there's three main types of muscle tissue that I want you to be aware of. And the pictures are shown here, so definitely know kind of like the differences in appearances of different types of muscle tissue. So to start with, we have smooth muscle tissue, and that's the picture on the top. And smooth muscle tissue, as opposed to the other two types, cardiac muscle tissue, and let me do that here so you can see that one, skeletal muscle tissue. If you look at those two, you kind of see some vertical markings going through the tissue, and those are called striations. And if you look at the smooth muscle tissue, well, you can see those dark spots, so that top figure, which are nuclei. You don't see any striations along the cells, so they're called smooth muscle tissue. And then, like I said, in the middle of each cell, you would have a nucleus, and then even though we kind of see the cells here all together, the ends of the cell pretty much like taper down. So if you look at your figure in your lab manual, which is on, I think it's the first figure that you have for exercise 25, so figure 25-1, that kind of shows you the appearance of smooth muscle tissue. And then, so where do you find smooth muscle tissue? So it's maybe something that we don't think of as much when we think of, saying something like a muscle. When we say that, we're usually referring to skeletal muscle tissue. Smooth muscle tissue is found lining your internal organs, and so even though we don't think about it, let's say for example if you eat like a hamburger, you swallow that, and it's not just gravity pushing your food through your intestines, it is contractions of smooth muscle tissue within the layers of the intestine that are kind of squeezing and moving the food along for you. Or another thing that is an example is, let's say with your heart is a different type of muscle tissue that we'll talk about in a second here. But in your blood vessels, especially in your arteries, you have layers of smooth muscle tissue that are contracting and helping move blood around, into like the various like extremities in your body. So that's smooth muscle tissue. So it's the muscle tissue found in your internal organs. Okay, and then so if you think about it, we don't have to think about it, right, to do those things. Like you don't have to voluntarily think about moving through your digestive tract. So smooth muscle contractions are involuntary, and they don't tire, so meaning they don't kind of tire in the same way that skeletal muscle, where you feel a sensation of you need to stop. That doesn't happen with smooth muscle tissue. Okay, then our next tissue type is cardiac muscle, and that's the picture in the middle in blue. So one thing you can see is it kind of has this characteristic like interwoven appearance to it. Like if you look at, so skeletal muscle is the one on the bottom, and if you compare it to that, skeletal muscle tissue is in kind of straight fibers, whereas cardiac muscle tissue has much more of like an interconnected appearance to it. The fibers are kind of branched, or the cells are branched, and we do see striations, so we see kind of like vertical lines on this tissue type as well. And one thing to think about as far as like, well, why are they kind of interwoven or interconnected like this, is that when the heart beats, that movement kind of has to occur in one single movement. And so the cells in cardiac muscle tissue are very interwoven because they're kind of acting as a single unit. Okay. Also, like smooth muscle tissue, it is involuntary, although, like we've kind of discussed before, there's a little bit of like a smooth line in between these definitions here, because you could think about it and slow your heartbeat down or slow your heart rate down, or you could be very nervous and your heart rate goes up. But generally, it's not something you have to consciously think about for it to happen. It is untiring, except for one fateful day, right? And that's always my joke. Okay, and then the last one is skeletal muscle tissue. So, like let's say you eat meat. You can kind of think of it as that is skeletal muscle tissue. It's the muscles that are associated with your skeleton. They're attached to your skeleton. So you can see that here, skeletal muscles are attached to our skeleton through tendons. And the things that are called ligaments, by the way, are the connective tissue that connects bone to bone. But what connects bone to your skeletal muscles are called tendons. So it's like a large kind of chunk of connective tissue doing that. And then kind of if you look at that cross section of muscle tissue, kind of think of that if you've ever eaten a piece of meat and you see it kind of has those kind of like appearance of like little fibers to it. Or if you think of like people pulling meat apart, you see those like individual fibers that it's made out of. And so when you get down to like the smallest unit or layer, each one of these kind of strands is one muscle fiber. Many of those connected together can make an even bigger kind of section, okay? So if you look at it here, so just kind of talking about, well, what makes up an individual muscle fiber? It's muscle cells kind of stacked end to end, and then they don't have any cell membrane between them, so they kind of look like one unit once they're fully formed. And you can see their nuclei kind of don't then have any like division between them. There's no barrier separating one cell from another, so the nuclei just kind of are there kind of on the end, so they can sometimes be referred to as like peripheral nuclei. Okay, we can see that vertical marking on these cells, so they're very much striated. Another term for skeletal muscle tissue is striated muscle tissue. It is definitely that it can tire as you work your skeletal muscles, they can tire out. And as we've seen, there are movements that are involuntary, for example, reflex movements that are movements of the skeletal muscle system that are involuntary. But generally, for the most part, we need to make conscious decisions to move our skeletal muscle tissue. Okay. One thing I'd like to point out is some of the work you'll do on your lab report. So I'm going to come back to that at the end of this section here. Okay, so let me go on, and I'm going to come back to exercise 25, but let me talk about this first. So we're going to move on to exercise 26, and I'd like to define a couple of terms for you. We're talking very generally about reproduction, and because this is a general biology course, we'll talk about a little bit more than just humans, but we'll focus on some human anatomy for reproduction. But here we're going to talk about some kind of like general patterns of reproduction that you find in vertebrate animals, so animals that would be considered chordates, but also you could apply this to things like some of the invertebrate groups that we've been talking about as well. And for that, I specifically mean these terms we have here. So first of all, we have fertilization differences. So not all organisms reproduce in a similar way to how animals do, how vertebrate animals do, or even mammals. We're talking about generally animals here, but not all animals reproduce in the same way we do. They can have very different means of reproduction. And you can start by thinking about, well, fertilization, that can be external or internal. So what do we mean by that? So external fertilization means that sperm and egg unite outside of the parents' bodies. So, this would be happening in the environment. So, for example, many organisms that live in aquatic environments are termed something called broadcast spawners. So things like sea urchins or sea stars or sponges or different cnidarians like a sea anemone. They think of like a sea urchin that's kind of, that little spiny ball, and they're just kind of crawling on the ground, or something like a sea anemone or a sponge where they're rooted to the ground, basically. They can move a little bit, but what they do is at certain times, let's say they're all of one, let's say we're talking about a particular sea star, and it's one particular species. That species will have a time that they release their gametes. So let's say the female sea stars, they release their eggs, and the male sea stars release their sperm into the water. And they have kind of chemical means of being compatible with each other, and fertilization will happen in the water. It'll form, let's say, a larval sea star, and they're just kind of independent, free-floating larvae. Usually they'll be part of like the plankton. So what that is saying is, you release your sperm and egg off into the environment. That's external fertilization. Then I have in the picture here an example where that would be true in vertebrate animals like frogs. So in these amphibians here, they have external fertilization too. And even though they kind of look like in this picture, like they're copulating, which would be kind of the act of like internal fertilization occurring, that's not what's happening here. What's happening is the female is on the bottom, the male is on the top, and the female is releasing her eggs, and the male is releasing his sperm at the same time. So the sperm and egg kind of meet, and it immediately goes down into the water. Like they're in this picture here, they're by a stream bank. And so the fertilized eggs kind of flood into the water. And so it's pretty different from how we would do things. So this would be an example in amphibians of external fertilization. Another group that it would be pretty common in are different fish groups. So, you can kind of think, for example, of like a salmon, and you see like the female salmon laying her eggs, and then the male comes over and releases his sperm over them and fertilizes them. So it's very common in the water that this would happen, and in fact, external fertilization, if you think about it, you're just releasing your kind of unprotected cells out into the environment. It has to be done in animals in a water environment. So in certain ways, for something like a fish, it's not really restricting them. But for something like an amphibian, it kind of means that their reproduction has to be tied to a water source. Like they have to be around a stream or they have to be around a pond if they do external fertilization. Another thing that you might not think about is, well, what does their egg need to look like? Okay, so you see here, I have a picture of some eggs in blue. So these are birds' eggs. These are robin's eggs, like you might have heard of the term like robin's egg blue. That's this color here. And if you think about it, if this female frog that I show you here was laying eggs that look like this, would the male sperm have any chance of fertilizing these if the eggs are encased in a hard shell? So the answer to that is no. It would be too tough, too thick of a layer for the sperm to actually penetrate through. So I want you to think about, like a fish egg. Like if you've seen like fish roe, they can be very small, they can be very delicate and very soft because they allow for a sperm to penetrate them, and this would have to be the case in organisms that have external fertilization, is you have to have a soft egg in order for that to happen. So even though, we're going to talk about things like reptiles and birds that potentially lay eggs, it's still that they would have internal fertilization because, obviously a hard-shelled egg, you cannot have a sperm penetrating that, okay? So in organisms and animals that have external fertilization, they have a soft egg that could potentially dry out, and so that's why it has to be tied to some source of water. Okay, so internal fertilization then just would mean, for example, that fertilization occurs, let's say in like the female's reproductive tract. It could be that you have various other types. If you had an organism that was like a hermaphrodite or something, could be different, but generally means fertilization occurs in the organism's body. So mammals would be an example of that. Birds would be an example of that. Reptiles. Some fishes would be an example of that too. And that is that the sperm is now protected because it's in like the male's either semen or in the female's reproductive tract. It's kind of all staying within the organism's bodies. It's never exposed to the outside environment. And so internal fertilization, in a way, allows a degree of independence from water as far as reproduction goes. Okay, and again, it just means that sperm and egg fertilize each other inside of, let's say, the female's reproductive tract. Okay, so then we go on to these vertebrate patterns of reproduction. So we have these three terms here, and they might seem a little dry to look at them, but we can kind of think about, well, what do they mean? And again, it's kind of an interesting thought as far as thinking about how other animals reproduce, and that it can be really different from how organisms like us, like mammals, would reproduce. So first, let me explain the first one, oviparous. So oviparous is basically an egg-laying organism. So that means that, and these patterns, by the way, these patterns of reproduction, they're distinguished by where does the embryo develop and how does the embryo receive its nourishment. Okay, so if it develops inside of an egg and it receives its nourishment from the egg, we would call that oviparous. And so oviparous organisms lay their eggs, meaning their eggs go out into the environment. So things like all birds, many reptiles would do this, many amphibians would do this as well, many fishes. Okay. And then I'm going to go on to the third term here, which is viviparous. Okay, and in this one, vivipary is the case, or would be the case in mammals. And what that means is that the embryo develops in the parent's body and receives nourishment from the parent as well. So in humans, that would be the case, that the embryo develops in the female's body and they receive nourishment directly from the female as well through an organ called a placenta. And actually you can see it here in this shark. So you see something like an umbilical cord connecting the mother shark to the pup, and it is an independent system of vivipary that's evolved in a few species of sharks, which is kind of interesting. Other organisms that do this, there's a few. There's actually a group of reptiles that do this, a group of amphibians that do this. They're called caecilians. They kind of look like a large earthworm in a way. They're pretty different from things like frogs and salamanders, but they're viviparous. And then there's actually an invertebrate group of organisms called velvet worms that are viviparous as well. So it's not just mammals that are viviparous. By the way, there's two groups of oviparous mammals, and I don't know if you remember those from our discussion on the chordates and mammals, but the platypus and the echidna are two egg-laying mammals, which is pretty interesting. Okay, and then we have this middle category called ovovivipary. So for this one, it's kind of an in-between of the two I've already mentioned. It's kind of interesting. So I knew when I was younger that sometimes rattlesnakes can have live birth. And I didn't really think about it too hard, and I just thought that it was, similar to mammal giving live birth. But it's actually pretty different. So in that case, what happens is the female retains her egg. So they do make an egg, but they don't lay their eggs. They remain inside of the female's body, which is pretty interesting. And you can think about, like, well, why would an organism do that? All right, why would they keep their eggs inside of their body? And what eventually happens is those eggs hatch inside of the female's reproductive tract, and then it appears like they're giving live birth. And in fact, the eggshells will be delivered after the babies are. And so in this case, in ovovivipary, the young, the embryos develop inside of the female's reproductive tract, but they get nourishment from the egg, not directly from the female's body. Okay? And again, going back to why, so it's actually like evolved a few times where in some snake groups In some groups of sharks, in some groups of frogs, we see this. And if you think about it, it's kind of like we're getting the benefit of not just laying our eggs out there in the environment, kind of vulnerable to predators. They're now retained inside of the adult's body, and it kind of adds a measure of safety because we can keep our young with us for as long as possible until they're kind of like in an older, less vulnerable state. So there is a lot of like kind of obvious benefit as to why something like this might happen. So ovoviviparous means the embryo develops in an egg inside of the mother's body. Okay, so for this one we can define it as development occurs inside the female's reproductive tract, but nourishment comes from the egg, right? In oviparous, the embryo develops in an egg and receives nourishment from the egg. In viviparous, the embryo develops in the female's body and directly receives nourishment from the female, the parent, as well. Okay. Alrighty, I'm going to go on. And so be ready for, let's say on like your practical, that you might see male and female reproductive systems. So we're going to go over some of the anatomy that we find in the male and the female reproductive tract. So this one, I apologize, is a little bit blurry. I'll show you a figure at the end of the lecture that looks a little bit less blurry, but this coincides with your figure 26-4. And it's on kind of the major anatomy of the female reproductive system. Let me go ahead and I'm actually going to go to this one here, and we'll start here because the way it's presented in the lab is kind of the path that sperm would take through the male and the female reproductive tract. So we'll start with the male. Okay, and as we go through this, we're going to go through the pathway, but we're also going to explain, or I will also explain some of the different glands that we find in the male reproductive tract. So they're not necessarily part, the ones that are numbered are the structures that are part of the pathway that sperm would take. The other structures are part of the male reproductive system, but they're not directly in the path that the sperm would take. Okay, so number one would be the testes. So you do have the scrotum, which is the skin surrounding the testes, but inside of that are the testes. And if you look at the picture on the upper left, that's kind of a depiction of what the testes look like. So they're a collection of tubes called seminiferous tubules, and inside of each one, if you look at your figure in your lab manual, and I'll tell you what that is. Let's see, in figure 26-1, it talks about spermatogenesis, and it shows you on the bottom of that figure a cross-section through a seminiferous tubule. So inside of each one of those tubes, you have cells that are going to become sperm, go through meiosis, a couple of rounds of cell division, and then some kind of modification to turn them into a sperm cell, because the sperm cell, remember, is kind of like the cell itself is very reduced. It's basically just the genetic information of the male. And then there's a tail packed with mitochondria, kind of acting as a motor to power the tail. And that's it. So all of that sperm formation is happening inside of the testes. Okay, so that's number one. Then number two is the kind of the beginning tube that I show resting on top of the testes. That is called the epididymis. And that is a storage region for the sperm. Then during a male ejaculation, the sperm is going to enter into the tube that's kind of next to the epididymis. And that is called the vas deferens. That is number 3. And if you follow it, the vas deferens goes from the scrotum and then up into the male's abdominal cavity. It rounds the bladder. And then it's going to meet up with a gland called a seminal vesicle. But up until that point, like right until it reaches the seminal vesicle, that is all the vas deferens, okay? And that is, by the way, like I'm sure you're familiar with the term vasectomy. So it's named after the fact that the vas deferens is cut during a vasectomy. It's cut kind of in the region close to the testes, so that the only thing missing from a male's ejaculation would be the sperm, which of course makes sense. And this is done in order to allow a man not to have a baby. Okay, so as we round over the bladder, and it meets with the seminal vesicle. So there's a tube coming out of the seminal vesicle and kind of meets with the vas deferens. So that area kind of after the seminal vesicle and right up into you have a junction with the urethra is called the ejaculatory duct. So that is number 4. So we have testes, epididymis, vas deferens, and then the ejaculatory duct. So that duct is coming from this gland called the seminal vesicle, okay? And once we kind of match that up with the bladder, so you can see the bladder is that large circle, all right, the one above the green circle, all right, that obviously would contain urine, okay? So in a male, the urethra has a shared feature, which would not be the case in a female. All right, so hopefully you realize that, although we will go over it here as well. All right, but after we go through the ejaculatory duct, we meet up with the tube coming out of the bladder. So therefore, we know that that is the urethra. Okay, and so that is the last structure in the pathway of the sperm in the male's body. Okay, let's see. Let's go through what some of these different glands are and what their functions are. Okay, so it's definitely the case that, like, the properties of semen, number one, like some of the questions kind of deal with this in the lab reports, to provide like a fluid medium for the sperm to swim through. But it's also the case that definitely with like a male's fertility, that it's very much determined by the number of surviving sperm. So the different glands produce things that are absolutely essential to keeping high numbers of sperm that eventually make it into the female's body. Okay, so one thing that's kind of interesting, for example, with the seminal vesicle. So what does that do? So that produces a secretion that neutralizes acidity in the female's reproductive tract. So let's go over here for a second. So if you look at the figure on the left, so that's kind of showing the uterus, and then kind of like the area that I have in blue, kind of going down to the outside of the body. That's the vagina. Okay, so you see that the vagina is this opening to the outside world. And so as far as we would go, like that area needs to be kind of inhospitable for things like bacteria, other just kind of microorganisms. So naturally in that area, there is a very high, or a relatively high, let's say, acidity level. And that's there as a natural defense against like a growth of bacteria. So like it's kind of like a natural system for keeping our bodies clean. All right, now the problem with that is that acidity is there to keep cell growth low. And so that would kill sperm if kind of just left that way. So what the seminal vesicle does is it produces a basic substance that neutralizes that acidity, and hopefully therefore, that increases the chances that when sperm enter the vagina, many of them are not going to die. Okay, the prostate gland adds carbohydrates to the semen, and remember, like I said, the sperm cell has lots and lots of mitochondria packed in it. That is where cellular respiration, or most of cellular respiration, occurs. And so a moving sperm cell is an energy dependent cell. So you're giving a fuel to the moving sperm through adding carbohydrates produced in the prostate gland. Then remember we talked about the fact that the urethra is shared with the excretory system, so there could be like remnants of urine in the urethra. So the bulbourethral gland produces a substance that neutralizes the urine that's potentially in the urethra and could be inhospitable to any sperm entering there. Okay, so then let's go through the female reproductive tract. So this is a little bit shorter, okay, and the figures here kind of just show like half versions of everything. The one on the right is showing the uterus, the oviduct or fallopian tube, and the ovary from a front view, and doesn't show the other side of that, but there would be another fallopian tube and ovary on the other side. It also doesn't show the vagina in this one. In the section on the left, we see it from a cross view, and you can see the vagina going into the uterus and then eventually into the fallopian tube. So again, we're just going to go through the pathway that the sperm would take, and we'll kind of talk about the different functions of the structures as we do that. So you can see too, and you can follow along on your figure. Again, this is figure 26-4 in your book. In front of your uterus and vagina are the bladder and the urethra. So those are separate from the vaginal canal and then the uterus, okay? So to start with, as far as the pathway that sperm would take upon entering the female's reproductive tract, the first place would be the vagina, all right? And then going on to the sections of the uterus, the second is actually the cervix. Okay, so the cervix is the entry to the uterus. It has several important functions. So number one, this is kind of like the doors that remain closed as far as like, you're not allowing anything from the outside world into the uterus. The uterus would be sterile. And so it has to remain fairly tightly closed, except at certain times. So, for example, during ovulation, normally that area has like a plug of thick mucus. During ovulation, there's kind of like areas, like little kind of like little tiny rivulets or rivers of area where the mucus is more liquid, and that would allow sperm to go through. But it's kind of trying to balance, allowing sperm in versus keeping bacteria and other microorganisms out. This is also the area, so if a woman becomes pregnant, this area does become sealed off, and it has to remain tightly closed. So, if you think about it going through the months of pregnancy and kind of the weight of the fetus's head on the uterus, that area still has to remain closed. Then during the time of delivery, that is the area that opens to 10 centimeters. That is the area that dilates. So like for example, with the vagina, that area just stretches as the baby's head comes through. But the cervix that is like during contractions, it is those muscles that are contracting and opening the cervix up. That's the beginning of the uterus. Okay, so beyond the cervix is the uterus. This is where a baby develops. And going past this, you can go into one of either tubes. So a woman would alternate in which ovary goes through ovulation each month. But basically, past the uterus, you go into the fallopian tube, and then the little pink dot I show is kind of potentially where most fertilizations would occur if that did happen. So it has to happen fairly high up in the fallopian tube. Once the egg has traveled all the way to the uterus, so much time has passed, about 2 or 3 days, that it's no longer viable. So fertilization occurs fairly high up in the fallopian tube or oviduct. And again, we would have 2 fallopian tubes on either side, like it would look bilaterally symmetrical. There's also 2 ovaries. Now, the ovary is not part of the pathway of the sperm, but that is where the egg was produced. And then every month, the ovary develops a cyst on it, and that bursts open. So when women feel like a pain during the month that's caused by ovulation, it's because it's literally that an area of the uterus kind of ruptures open to allow the egg to release from it. Then there's kind of like little fingers on the end of the fallopian tube that sweep the egg up into it. And then eventually that moves down, and if it was fertilized, it'll implant into the uterus. Okay? So pathway for the sperm in the female's reproductive tract, vagina, cervix, uterus, and then the oviduct or fallopian tube. Okay, so lastly, we're going to talk about genetics. And I'll see what we're going to do as far as how the lab handles it. It's kind of fun, the stuff we do in genetics as a group. So potentially we'll still do this together as a group, but we'll see. Okay, so we're going to talk about a lot of different terms in this lab, but I also hope that you can kind of pull like what the underlying concepts are from these, because it's actually pretty interesting. And I like to kind of explain things through analogy because I think that gives us kind of a frame of reference for our learning. So I think as long as you take it for what it's worth kind of a thing, I think analogies can be a really good kind of tool for introducing you to something. So first we're going to talk about these two terms, genotype and phenotype. Okay, and I have these different characters here to kind of model and be my examples. If you kind of think about an organism, kind of like the whole organism here, like let's look at like the little girl in the middle of the picture. Any organism, but this little girl included, is like a collection of traits. There are different traits that make up her physical body. Okay, and that's kind of like my analogy would be if you think of like a house. Like a house is a physical structure, and it's made up of different structures, right? Like it has an exterior, let's say it's made of brick, but then it also would have plumbing, it would have electricity, it has these different systems, right? Kind of like we would have different organ systems working together for a common function, okay? So when you build a house, usually it's the case that you have something called blueprints for those. And each different page will kind of specify different things, like maybe the exterior, like use this type of brick, put this window in this location. Then there's a page for the plumbing, then there's a page for the electricity and where everything goes and what to use to do that, okay? And so those two things are kind of like the difference or the distinguishment between genotype and phenotype. Okay, the phenotype is the observable traits, like the physical traits of an organism. So the phenotype in the house is just the actual physical house and all of its components that it's made of. It's what it is, and we can kind of break those down into different parts, right? Like we can say this particular pipe of this particular diameter, and it's made of copper, or it is made of 4 walls on the outside that are made of brick. So we're describing different attributes of that house, right? Well, in an organism, we would call those its physical traits. And those physical traits, for the most part, like definitely when we talk about it in the context of learning about genetics, we talk about that they're under the control or the influence of your genes, right? And that goes to, in the same way that that house, the final resulting house was completely affected by those blueprints, right? The blueprints kind of dictated how that house and how the structure and the components of the house eventually turned out. And so if we think of our genes as our instructions for how to build us as organisms, that's what they are. They are the blueprints. They are the instructions that say, put in this particular type, or put in this other particular type. Structure this in this way. It encodes for all of our traits. And so our genes are the actual genes that we possess. Okay, so that's our genotype. Our phenotype is heavily influenced by our genotype. Our traits come from our gene expression. Now, they also come from other things too. Like let's say we had the same blueprints and we built 2 different houses, okay? They're going to turn out slightly differently. Or let's say one house was built in Alaska and one house was built in Florida. over time, let's say the exterior brick of a house is going to look slightly different because they're in two different environments, right? So our traits are a mixture of the genes that gave instructions on how to make those traits, and also the environment that kind of affects the traits as well. Okay, but it's definitely the case that our traits are heavily under the influence of our genes, and definitely some more than others, right? But in this context, what I want you to know as far as what does genotype mean and what does phenotype mean, the genes, the genotype are the genes that we actually have, and then the phenotype is the result of that gene expression. Okay, so we've been talking about it pretty generally, but I'm also going to talk about genotype and phenotype in the context of this example here. And we'll talk about some other things as well. So if you think back to mitosis, and we talked about the fact that we have homologous chromosomes. Remember, with humans, as an example, we have 46 chromosomes, but the actual information needed to make a human being is contained on 23. We just have 2 copies of every chromosome. All right, and those are called homologous chromosomes, and we get one from our mom and one from our dad. Okay, so what that means then is on those homologous chromosomes, they're going to have similar genes. Okay, like again, we have 2 copies of every chromosome. So for example, let's say on chromosome 15, you have a gene for eye color. Okay, that means you have 2 chromosomes 15. So you have the eye color version that you got from your mom, and you have the one you got from your dad. Okay, so everybody has that. So in my example here, I have Padme and Anakin, their mom and dad, and then I have little Princess Leia. So mom and dad both have 2 genes for eye color as well, and then so does she. But then in this example, I show that Padme passes on a gene for brown eyes, and Anakin passes on a gene for blue eyes. Because remember, we go through meiosis and we reduce our chromosomes down by half. Okay, so in any sperm and egg cell, there are 23 chromosomes, okay, not 46. So when they come together, we go back to 46, okay? So that's kind of an important point too, is our parents only give us one of their copies of their genes that they possess. For each gene, that would be true. Okay, so we still get a complete set from our parents, but it's one of each gene that we would possess. They come together and they result in a person that has 2 copies of every gene. And that's always the case, all right? So we're looking at it here with just one example, okay, that Padme donated a gene for, let's say, brown eyes. Anakin donated a gene for blue eyes. Excuse me. Okay. So, in talking about genotype and phenotype then, okay, so we can describe the girl's genotype as she has a gene for brown eyes and she has a gene for blue eyes. Those are the genes that she possesses. And you can kind of think of it as saying she has instructions for brown eyes and she has instructions for blue eyes, right? Because that's what our genes are. Okay, so let's talk about a few other things here. So she has a gene for blue eyes and a gene for brown eyes. So generally, you could say that we have an eye color gene. And by the way, I'm kind of simplifying it a little bit. Like there's more than one gene that ultimately results in our eye color. But in general, our genes work this way at the level of single genes. So let's say, for example, we have a gene for eye color. Okay, but we can see here in her, she has a version that's going to give her brown eyes and a version that could potentially give her blue eyes. So genes in general kind of come in different versions. All right, so there can be alternate forms of our genes, even though like the category remains the same. their ultimate result could be slightly different, and those are called alleles. So when you hear the term allele, just think of the phrase form of a gene. So if I say the brown allele, that's the brown form of the eye color gene. The blue allele is the blue form of the eye color gene. So it's kind of that we have the same category, but then there can be slight differences between them. Not all of our genes are this way. Some of our genes are, they come in one form and that's it. Especially things that are like super, super essential, like the genes that like encode for the enzymes involved in cellular respiration. A lot of the times they don't vary that much because they're kind of so essential, and any kind of differences would just result in something that didn't work. But something like eye color, you know, if there was variation, like a mutation that popped up that gave a different eye color, that would kind of be neutral or, you know, potentially neutral, right? It's not these like immediate negative consequences. So for a lot of traits, a lot of things about us, that would be the case. And so for a lot of genes, they come in all of these different versions or different alleles, okay? So if I said her genotype is big B, little b, that is a description of her genes she possesses, her genotype. And in fact, let's say it was the case that for eye color, there are these two alleles, these two forms of the gene. Big B gives brown eyes. Little b instructs for blue eyes. So there's two alleles for the eye color gene in my example here. So we can see the other thing too, illustrated by her and her parents, is that because of that, any potential person's genotype then can kind of come in three versions, right? Like you can have someone that had only big B, you can have someone that had only little b, and you could have someone that was a mixture of the two, okay? So then I'm going to throw in a couple of other terms here, okay? Now, actually, before I do that, let me back up a second. So let's say with Padme, okay, she has a gene for brown and another gene for brown. That's her genotype. But then if you look at her phenotype is that she has brown eyes. That's her trait. That was heavily influenced by the fact that she has two genes for brown eyes, if not like almost completely all influenced by that. Okay, like eye color would be a heavily genetic trait. Okay, and then in Anakin, I'm not exactly sure what color his eyes are, but let's say he has blue eyes. He has a gene for blue and another gene for blue. Therefore, he has blue eyes. So again, his genotype is little b. His phenotype, his trait that he has resulting from his genotype, is blue eyes, okay? So the phenotype is a description of your traits that you actually have that come from, or at least heavily influenced by, a particular genotype, a description of the genes that you actually have. All right, now, let's say we didn't know what a combination of those two different genes resulted in, but we saw that in their offspring, she has brown eyes. Okay, so what that means is that if we know that she is big B, little b, she has one of each, and it happens that she has brown eyes, then that's telling us that brown is the dominant gene. Okay, if she had ended up with blue eyes, that would tell us blue is the dominant gene. If she has one of each, but in her ultimate trait, she has only one of them, that is describing a dominant recessive relationship in the genes. Because if you think about it, it's that your genes, if you have two different ones, they can kind of interact differently. There are more than just dominant and recessive relationships. There are other ones, but we're just going to talk about this one. Okay, and so a dominant gene is one that if it's present in your genotype, you're going to have that, the result of whatever that gene is. You're going to have it in your phenotype. So for example, big B is the gene for brown eyes. So if you have it, you're going to have brown eyes. Like you can see, you can have both, or you can just have one. So if it's there, you will have brown eyes. And then kind of the reverse is true for recessive. The only way you're going to have the recessive trait is if the dominant gene is not present. Okay, so the only way you have the recessive trait is to have two of the same recessive gene, so little b in this example. Okay, and then again, to kind of throw some terms out there for you. So there's terms that describe these different states of our genes. Okay, so if you have two of the same genes, that state of being like that is called homozygous. Okay, so that's what the parents are in this case. And then the offspring, because she has two different alleles, two different forms of the eye color gene, she is heterozygous. So homozygous and heterozygous, those are descriptions of your genotype, the genes you actually possess. Okay? Now we can go even further for this homozygous term. So if you are homozygous, meaning you possess two of the same forms of the gene, two of the same alleles, you could be like Padme and have two of the same dominant alleles, so she is homozygous dominant. Okay, and Anakin is therefore homozygous recessive. All right, so if we go from left to right, Padme is homozygous dominant. Little Leia here is heterozygous. And Anakin is homozygous recessive. All right, so those terms are descriptions of the genotype, and if you hear them, you should kind of have an idea. Like, we could interchange any trait in here, but they would still be, you know, two of the same or two different. Okay. Alrighty, so let's see. I'm trying to think if I missed anything here. Let's see. Okay, I think that's all. I think that is all I want to talk about with you guys as far as kind of like the bigger picture. What do these different terms mean? Okay, I have this other picture here kind of written out and, you know, kind of describes everything here, some notes here for you to kind of look at. All right. I can go on to talking about what it means to have different sex-linked traits, so I'll do that as well, and hopefully that will help you to finish your lab report. Okay, so you might have heard of, like, let's say for example with color blindness. You might know if I asked you, if I said someone came into my room and they were colorblind, and I said, what biological sex do you think they were? You might answer male. Okay, like that is kind of something that's commonly known. So color blindness is something called a sex-linked trait. Because it's linked to our sex chromosomes. All right, and in humans or mammals in general, if your chromosomes are XX and there's no malfunctions going on, you're going to end up a biological female. And if you are XY and there's no other malfunction going on, you're going to end up a biological male. Okay, and if we look at the chromosomes, you'll see, number one, hopefully you notice the difference between the two. Okay, like notice that the Y chromosome is much, much smaller than the X chromosome. And if we look at what I have written here, the Y chromosome holds only a dozen or so genes on it. And they have to do with sex determination, and which is a very interesting topic at a certain point in our development. Up to that point, we look the same. Male or female have the same structures. But then at about 2 months of development, if you have the Y chromosome, there's genes on the Y chromosome that are going to start expressing and start turning the anatomy of that fetus into a male. Okay, and that's basically all of the genes are like that on the Y chromosome. They just code for sex determination. But the X chromosome, that's not the case. It holds many, many genes, 2,000 to 3,000 genes, and a lot of them have nothing to do with sex determination. So anything that is a sex-linked trait would be a trait that's found on the Y chromosome. Okay, it would be a gene that's found, on the Y chromosome. All right, so our ability to see color, all right, those genes are found on the X chromosome. So now if you think about it, so let's say for example with color blindness, all right, actually let me go to this figure here really quick. Okay, so we can talk about this with color blindness, but also kind of with baldness is also something that has different results in male and female. It's a little bit more complicated than just our genetics, or it also has to do with the fact that males produce higher levels of testosterone than females. But other examples of sex-linked traits, and this is kind of like more when the traits go wrong, is hemophilia. So the trait that would be linked to this would be kind of like our ability to produce the usual levels of platelets in our blood. Albinism, which would have to do with like malfunction of melanin production. Myopia, which has to do with our vision. And then the other one is detection of color. So we have cells in our eye that detect color, and you have genes that encode for certain pigments that allow us to discern different colors. So for example here, like I have this picture here, which is a color blindness test. So I believe, if you look at it for a second, even though it's a little not super obvious, I believe there is a number 5 in there. So if you're colorblind, everything would just look the same to you. So it doesn't mean, now there's different levels of color blindness. It would kind of mean that they would see less colors in the rainbow. They don't see necessarily in black and white, although there can be color blindness that is like that. But usually the most common one is red-green colorblind, where the person would lack the ability to distinguish between the colors red and green. They would look the same to them. And just for your information, there are animals that have more rods and cones than we do and can see more colors than us. So that's kind of interesting too. Like they would see more colors in a rainbow than we would. But back to this. Okay, so if you look at the numbers here, so 8% of males are affected by color blindness, where it's only 1% of females. So it can happen in females, it's just much less common. Okay, so going back to this one here, oh, and there's some other ones you can kind of use just as color blindness tests, by the way. But let me go back to this one here. Okay, so what you can imagine then is, let's say, for example, the genes for producing the normal pigments or the gene for producing normal vision has a malfunction. All right, and that malfunction is going to result in color blindness. But I'll also say that it is recessive to normal vision, or the genes for normal vision. So now let's imagine you were a woman and you had one normal copy, but you inherited a gene for color blindness. All right, because color blindness is recessive, if you have a normal copy, the normal copy is going to cover for that malfunctioning copy, and you won't be colorblind. But now let's imagine you're a male and you inherit one gene for color blindness. You would have it because you don't have another copy. So for the sex chromosomes, males and females are slightly different. Like females are diploid. They have 2 copies of the X chromosome. But males, for the X and Y chromosome, those are 2 different chromosomes. So for the sex chromosomes, males are not technically diploid. All right, and so if there's any gene that's malfunctioning on the X chromosome, then that person is just going to have that that result. They're going to have that disorder in the actual person, right? If they're male, okay, they only need 1 copy in order to have the resulting disorder, whereas females need to have 2. Okay, so I can go on to help you with this potentially in like a Zoom meeting, all right? But that is kind of like the background explanation for why it's more common in males to have a resulting disorder if it's a sex-linked trait
than in females. Females have to have 2 copies of a malfunctioning gene, where males only have to have 1. Okay, so I hope that helped you guys. I might add a little bit more onto this just to kind of go over some of the different structures in the lab report. But other than that, hope that helped you guys, and see you soon.